So good afternoon, everyone, and welcome to this joint EuroCC and CIMEA online seminar. Today our speaker is Professor Vangelis Daskalakis, who is the head of the Computational Environmental Modeling Group in the Department of Chemical Engineering of the Cyprus University of Technology. His research focuses on the biophysics of large biological and small gaseous molecules involved in processes related to life sciences and the environment. He uses elaborate computational techniques based on molecular dynamics in combination with ab initio density functional theory methods. Professor Daskalakis is an established HPC user and has been awarded computational resources on the national HPC system, as well as other European and praised systems. Today, he will present to us in this seminar some of his work, which is titled High Performance Computing in Structural Biology. So, Professor Daskalakis, if you're ready, please start. Thank you very much.
Yes. Hello. Thank you very much for the invitation to present part of my work in structural biology and how this was made possible by employing high-performance computing. Vangelis Daskalakis from the Department of Chemical Engineering and Cyprus University of Technology. So, no movement, sorry. Okay, so some introduction on the tools we use to study structural biology and protein structure and function. We know that structure is the basic element to understand nature. The structural details are important to admire nature, as you can see here. However, not all information is stored within a structure. To find a final conformation, get from an initial conformation, you need the dynamics. The dynamics are important to walk you through the initial conformation of a protein through the final conformation, which is also important for the function. So our approach in the lab to study protein structure and function is based on the motion of atoms and molecules. Protein atoms move on free energy surfaces like these here between energy minima, the red areas as you can see here. The atoms move and convert energy for biological work. This is a definition of bioenergetics. So we have flexible proteins with many minima, red areas in the free energy surface, many conformations, different structures of the protein, and freely moving atoms. However, we have also rigid proteins with a few minima in the free energy surfaces, small number of conformations, and confinement in the atom motion. So in the lab, we start from a protein structure. We can change a protonation state of the structure. We can mutate residues. We can add a membrane, and we can solvate. And this is a start of an approach called molecular dynamics, which has a simple theoretical basis. If a force F is applied on a mass m at position x0, then this mass is moving to position x1 over time dt and obtaining velocity v, based of course on Newton's equations of motion. That is how we also move atoms in proteins. We have an initial structure, as I told you, based on crystallography or predictions. We convert it to a free energy surface, and we can move from a structure 1 of protein to a structure 2, a conformation 1 or conformation 2. If we know the force which is applied on every atom, which is calculated based on the derivative of the potential energy surface, and we can get the trajectory of atoms. This means that we can have a protein moving, the protein atoms moving, and the protein function like we could observe it under a powerful microscope, of course in correlation with experimental work. We have the system, the protein, usually solvated with also ions. We have the simulation cell with the protein within, the water molecules around, and we can decompose this simulation cell in several domains. Each domain is taken care of a number of CPUs in high performance computing systems. Usually our systems contain around 200,000 atoms per simulation cell, and we use around 256 standard CPU cores per simulation cell. Usually the simulations we're performing need like 9 cell replicas like these, and this amounts to over 2,000 standard CPU cores per run. Structural biology meets molecular dynamics in many publications in international journals, like this high-resolution structure and molecular simulations of a key bioenergetic protein from the complex one from respiratory chain. Or the simulation of an entire cell organelle, as you can see here, a photosynthetic chromophore vesicle from purple bacteria at all atom resolution. On the lab, we concentrate on the processes of oxygen cycle, which is related to photosynthesis, which is the absorption of energy from the sun, and cellular respiration, which is related to oxygen reduction. These processes are also related to decrease in carbon dioxide, atmospheric carbon dioxide concentration over spring and summer, and increase of this carbon dioxide concentration over autumn and winter when the photosynthetic machinery is at dorm. Cellular respiration or photosynthesis are essential to most aspects of cellular metabolism, therefore to life itself. There are several publications in journals like Nature and Science based on this research. So it's a hot subject and an active field of research. We concentrate, as I told you, on photosynthesis and cellular respiration that rely on protein complexes to harvest and convert energy. Remember that I told you that proteins can convert energy through the motion of their atoms. So we rely on 2 complexes, actually Photosystem 2, which oxidizes water to oxygen, and cytochrome C oxidase, which reduces oxygen back to water. And these processes are coupled to anabolic and catabolic metabolic pathways. This talk will be concentrated on Photosystem 2 and the related proteins. Structural details of the energy conversion within these proteins are missing in the literature. So we need information on how we can get from a protein structure 1 to a protein structure 2 related to photosynthesis, let's say. A small introduction to photosynthesis. Antennas or light harvesting complexes, they call it, around a super complex called Photosystem 2, absorb light energy and direct this energy to reaction centers in order for the oxygen evolution to take place and oxidation of water, as I told you before. However, in bright light or excess light, the absorbed excess energy is converted to heat and lost, and it is not directed to reaction centers. This is to protect the photosystem 2 apparatus. Therefore, there are 2 states of the light harvesting complexes around Photosystem 2. There is the light harvesting states, which absorb sunlight in normal light conditions and direct it to the reaction centers of Photosystem 2. And there is the photoprotective state, where energy in excess is absorbed and not directed to reaction centers, but dissipated as heat in the environment. Unfortunately, there is no crystal structure yet for the light harvesting conformation, and this is what we're looking for. There is a global interest in looking for that structure, the light harvesting structure of the antenna system of photosystem 2, because in bright light, the absorbed energy from the sun is lost, as I told you, as heat, and the biomass production is severely reduced compared to the biomass produced under normal light conditions. Also, there is, of course, an active presence in the literature, in journals like Science and Nature, with researchers looking for that structure. And why, again? Because we can also develop artificial photosynthesis systems based on the light harvesting structure of light harvesting complex 2. Also, we can have tolerant plants and increase of biomass if we can control these photoprotective mechanisms of plants, especially in harsh conditions of soil, water, or temperature. So again, this is an active field of research, and many groups worldwide are interested in this research. But first, we need to resolve and find this light harvesting conformation structure. In vivo, the transition is achieved by the interaction with a protein called PSBS. The light harvesting complex is interacting with a protein called PSBS. This interaction leads to aggregations of both light harvesting complex 2 and photosystem subunit S, as it is called, this PSBS protein. However, the details at the atomic scale are unknown, and we need molecular dynamics and computations to figure out how this transition is achieved, and if actually we can guess the light harvesting structure of photosystem 2 antennas. Our model is the antenna of photosystem 2, which is a trimer, the green protein, as you can see here. It is embedded in thylakoid membrane or a liposome. And PSBS protein, the photoprotective PSBS protein, is around this light harvesting complex 2 trimer. We have accurately predicted the position of PSBS protein around this light harvesting complex trimer based on the actual crystal structure where it was absent. And we monitored the antenna aggregation. The antenna is the green protein here. In the absence and the presence of the PSBS protein, the gray protein, as you can see here, as we would watch the process, the whole process on a microscope. So we have monitored changes in the shape of the light harvesting complex 2. That also induces changes in the thylakoid membrane, the thickness of the thylakoid membrane under circumstances of photoprotection. And this was also experimentally proved. The sampling duration for molecular dynamics simulations was around 150 microseconds, while the literature state of the art was at the moment only 14 microseconds. So 10-fold increase in sampling enabled by high-performance computer resources. So we managed to generate a prediction for the light harvesting conformation of the major antenna photosystem 2 and how it changes towards the photoprotective mode as the arrows depict here. This is a small video on the changes of the light harvesting complex upon induction of the photoprotection. So we were able to identify the elusive light harvesting structure with large chlorophyll excited state lifetime, which means that it gives time for the chlorophyll to transfer the energy to the reaction centers of photosystem 2. And it is not dissipated as heat. And of course, we have initiated targeted experimental studies on mutated residues, the residues you see in blue here, which are important for the transition between the light harvesting and the photoprotective state of the antenna of photosystem 2. This project was based on the PRAISE scheme, and we got awarded 17 million standard CPU core hours for 12 months on the supercomputer SuperMUC NG, which consists of around 300,000 computational cores and 26.9 petaflops per second. Now moving to another project related to coronavirus, the new coronavirus. It is based on a collaboration between Cyprus University of Technology, myself, and the University of Crete, and professors, colleagues from the School of Medicine, the Department of Endocrinology, Clinical Virology, and also the Department of Biology in the University of Crete. A small introduction, we know that viruses express lots of proteins that help in propagating the virus and replicating the virus and for viral infections. We are interested in two specific proteins. One is called 3CL-Pro, which is a protease. It cleaves an inactive polyprotein of the coronavirus into 11 functional viral proteins, and it is active as a dimer. Also, we target another protein called nucleocapsid of the coronavirus, which packs and imports the coronavirus viral RNA into the host cell nucleus for the infection. It also acts like a dimer and interacts with proteins called importins, which enable the entering of the viral RNA into the human cell nucleus. What we did is we tried to translate these proteins into digits and then use molecular dynamics simulations to see how the dynamics of these proteins and how we can interfere with these dynamics to stop the viral infection. We targeted these proteins with natural products that could act like antiviral drugs. And these natural products were targeting the dimerization of the new coronavirus main protease and how this dimerization is destabilized in the presence of natural products. And also the interaction of the nucleocapsid protein of the coronavirus with importins of the host that pack, as I told you, and transport the viral RNA into the host cell nucleus, and how this interaction is interrupted in the presence of natural products. And by interrupting this interaction, we stop the actual viral infection. Now, based on these results, guided experimental studies are taking place in labs, in foreign labs that are certified to handle coronavirus samples. Some details on this study. We found out that the main protease dimerization is destabilized by the binding of a natural polyphenol called fortunelin, while the binding of PCMN to the nucleocapsid protein of the coronavirus destabilizes the interaction with the importins and the entrance of the viral RNA into the cell nucleus. We have found also important residues of the nucleocapsid proteins for this interaction and this disruption upon binding to natural products. but also important residues on the binding of fortunelin to the main protease of the virus. And luckily, these residues are not mutated in the new mutations of the coronavirus. This is a scheme of the main protease of the coronavirus. And this is the place where fortunelin binds, the natural product. Based on these molecular dynamics simulations and the free energy surfaces we could produce out of these dynamics, we saw that we realized that the binding of fortunelin weakens the dimerization because one important structure of the main protease dimer is absent in the presence of fortunelin. This C1 structure here is missing, which is the stable structure of the main protease dimer, which induces the infection, cuts the polyprotein into 11 active viral proteins. Now moving to details on the nucleocapsid protein and the interaction with P-Simon. This is the whole nucleocapsid protein. It's a large protein. We only concentrated on this side here, and where we could bind the P-Simon, which can be found in cumin or thyme, and see how the interaction with important proteins like importins, which, as I told you, pack and direct the viral RNA into the human host cell nucleus. We could see that P-Simon interacts with coronavirus nucleocapsid protein, impairing the interaction with importin. This is a stable nucleocapsid importin interaction, and upon the binding of P-Simon in this complex here, we move to the structure M2, which is, of course, where the interaction between nucleocapsid and importin is severely weakened. And this is also shown on the free energy surfaces produced for this study. We have a barrier and a larger barrier to go from structure Mi1, which is M1, which is a stable complex of importin and nucleocapsid, to the dissociation of nucleocapsid from importin. However, we have a very low barrier in the presence of P-Simon, and this structure here is destabilized and moves to this conformation, which is, of course, not active for viral infection. Another aspect of this study was to produce residue sequences that could be used as vaccines against the coronavirus. We identified, based on the dynamics I showed you earlier, we identified several regions on the nucleocapsid protein that could act as epitopes. Epitopes are small regions of proteins that are recognized by antibodies and induce an immune response. So we identified important regions of the nucleocapsid protein that, if present as a vaccine and injected in a human being, it could produce an immune response and shield the individual against the coronavirus. This project was allocated 18 million standard CPU hours for 6 months on a supercomputer, Irene-Rome, which has around 293,000 CPU cores and 11.75 petaflops per second peak power. This is the description of our work in a magazine called Praise Digest, which hosts several successful projects on high performance computing projects. By now, it is clear that molecular dynamics with new sampling techniques and high-performance computing resources can lead to accurate description of the structure and function of large protein complexes at atomic scale, contributing essential knowledge in structural biology. Simulation outputs at atomic scale exert consistency with the experiments and can guide experiments with considerably lower costs. This research on bioenergetics of photosynthesis, some research on bioenergetics of respiration have produced several publications from my group, like 35 publications. But also we plan to expand to experimental studies guided by these simulations, like spectroscopic studies in correlation with ab initio computational methods for the structural changes in the light harvesting complex 2 antenna, simulations and experimental imaging of the effect of cholesterol, plant sterols, and exchange of proton deuterium on the structure dynamics of the light harvesting complex 2, photosystem 2. Also, a project on diatoms, which are responsible for the 20% of the global carbon dioxide assimilation. Of course, applications in artificial photosynthesis with a large, complex and model of photosystem 2 and LHC2 antenna of millions of atoms in, like the publication in Cell and the organelle I showed you in the introduction, with collaborations from foreign universities and colleagues from Cyprus University of Technology. Of course, I have to thank the research funding, which is mainly based on PRAISE scheme. With over the last 2 years, it is around 48 million standard CPU core hours, which is translated to around 2.7 million euros, with 2 huge projects, one on COVID and the other on photoprotection. And of course, the funding from Cyprus and startup grants and networking for the human resources and equipment. Thank you very much for your attention and again for the invitation and the opportunity to present my work, which is based on high performance computing.
Thank you very much, Professor Daskalakis, for your talk and your presentation of your work. I'd like to ask the audience if they have any questions to ask you concerning your presentation. I do as well later. Professor Alexandrou, first, please.
Yes, thanks very much, Vangelis, for this very nice talk. If I understand correctly, you're using software that run on CPUs, right?
Right, yes.
As you know, we will have now big developments that would involve GPU computers like the Lumi in Finland.
Yes.
My question is, are you also involved in code development for different architectures? Because this is wonderful to see your success on the European scale, and I hope our government will see this as well. But going forward, what are your plans?
The codes are already imported to the GPU architecture, so we haven't tested actually the code on GPUs, but there is the possibility to test the code on GPUs, and we would like to test it, and because I think we would have a great acceleration on our simulations. However, I guess that the transition won't be smooth, but it will be fruitful, for sure.
Okay, maybe you can talk more about this, Vangelis, at some point.
Yes, sure.
Thank you very much. Very nice talk. Thank you.
Thank you.
Kathy, I think you were next.
Good afternoon, everyone. Firstly, I would just like to say thank you very much for the presentation. We really enjoyed both projects. The one question I wanted to ask relates to the library of natural products that you used in your study. If you could just elaborate on that, I was just wondering, are these natural products that were tested against COVID, or is it just a natural product library of drugs that are currently on the market? Could you just elaborate a bit on that?
Yeah, sure. It's called Zinc database of natural products. It's not specialized in drugs or let's say dietary supplements or something like that. There are natural products like from fruits, from vegetables, from plants. It's a huge database on natural products, not specifically to be used like drugs or something like that. So it's not specialized, but it's a huge database.
Okay, thank you very much.
Sorry, Kathy. I had a sort of similar question when I saw your presentation. Did your results find that a specific enzyme or molecule of inside cumin inhibits the spread of?
Yeah, the P-Simon, it's the compound.
So we
it's
should.
Called. I can go. Yeah, this is the structure of the compound, and some analogs also work as potent inhibitors of the viral infection. But of course, these are initial simulations. We are now conducting experimental studies to make sure we have the correct, let's say, food supplements.
Okay, good. So I think it was Banayota next.
Hello, thank you very much for a very stimulating talk. I was wondering whether you could elaborate on the variation in thickness of the membrane, the liposome membrane, and the first part of the talk. Is that part of how important is it, and is it like a filter of the light?
Yeah, okay. What we have found out is that when this antenna of photosystem 2 is changing shape, the membrane becomes thinner, which is also observed experimentally. And we believe that this thinning of the membrane is caused by a process called hydrophobic mismatch. Because of this shape change of the antenna, some thylakoid lipids are concentrated in this area to let's say, cover for the hydrophobic mismatch. Hydrophobic mismatch means that there is a mismatch between the hydrophobic area of the membrane and the hydrophobic area of the protein. Usually, you need to have the same thickness of the membrane should be reflected to the thickness of the hydrophobic region of the protein. If there is not this one-to-one mapping, then there is a mismatch. So let's say protein gets bigger than the hydrophobic region of the membrane, or the membrane gets bigger than the hydrophobic region of the protein. Due to this hydrophobic mismatch, some lipids are concentrated in this area. So we believe that this induces a thinning of the thylakoid membrane, which is also inducing the aggregation of the antenna complexes, because imagine that you have 2 drops of olive oil in a glass of water. They will get together. So that's how we vision this mechanism of aggregation in the presence of the light harvesting complex under photoprotection.
Thank you very much.
Mm-hmm.
Professor Hartmann-Laris, you have your hand up next.
Yes, thank you. Thank you very much for the nice talk and nice overview. That's a more general question. I'm wondering about the convergence of the free energy surface plots that you presented in several cases.
Mm-hmm.
If I understand well, you use a replica exchange, more or less, a standard sampling technique.
Yes, we use the replica exchange. We have performed many enhanced sampling simulations. So it's not actually only replica exchange. It's also multi-Walker metadynamics. It's simple metadynamics. So we tried to apply every known enhanced sampling method. That's why we got this 150 microsecond sampling. We had the time, the computational time to do actually that. So we had the time and the means to apply every enhanced sampling method. And we saw actually that, okay, the results are convergent and we have actually a result that we can rely on from different methods.
So if you use then, just a follow-up question, let's say, if you use metadynamics, what is the order parameter that you use in your case? Is it straightforward? What is the correct order parameter or not?
Okay, so that's let's say a technical question to answer, but what we do is actually run long classical molecular dynamics trajectory, very long, and then use Markov state modeling to, let's say, to identify the important degrees of freedom, actually the slowest degrees of freedom between changes in the shape of light harvesting complex. So out of these slower degrees of freedom, we construct two collective variables as a linear, let's say, linear sum of torsional angles of the residues involved, the important residues involved in this shape change. So this is our order or collective variable parameters. So it's a little bit complicated as a coordinate.
Just wondering how, I mean, whether you can check, let's say, do a double check and further examine whether this is the correct order parameter. But okay, this is
we tried, of course. We saw that different methods converge to the same free energy surfaces, and also we identified that two different states, C1 and C2, that have a distinct excited state chlorophyll lifetime, which means that the conformations we, let's say, distinguish and separate in this free energy surface are one belonging to the light harvesting structure and one to the quenched state. That was our aim, to just able to distinguish between these two states. Now, as far as the validity goes for these results, we need the mutants and see if we mutate special residues that are involved in these coordinates of the free energy surface have any impact on the, let's say, photoprotection of the plants.
Okay, and if I may add just a relevant question. So at some point you mentioned that you also perform or you collaborate with some experimental people. So are you performing experiments on the systems as well?
Yes, yes, sure. Yes.
Okay.
Raman imaging and I can show you. Yes, it's further away. Yes, we have collaborations to start performing, to have some spectroscopic studies in correlation with ab initio computational methods for these structural changes. Some imaging techniques like freeze fracture electron microscopy and super resolution microscopy to see how, because in the process of photoprotection, there is also a pH-dependent mechanism. So we also probing these, and also the effects of cholesterol or plant sterols on the thinning of the thylakoid membrane, because the approach is twofold. We have some changes happening on the thylakoid membrane itself and some changes happening on the LHC2 structure. The changes on the LHC2 structures are mainly dependent on the pH, while we can control the changes on the thylakoid membrane by the amount of cholesterol or plant sterols we introduced in the thylakoid membrane. So we can correlate our experimental findings.
That's very interesting, because from this, for example, if you have Raman spectra, then you can also calculate the Raman spectra from the
we have Raman spectra on the carotenoids, because of course I could not show everything here, but there is an interaction between a chlorophyll and a carotenoid that makes possible this transition from light harvesting to the photoprotective state. So we can monitor the structure of carotenoids within the light harvesting complex by Raman imaging. And we're in the process of doing these experiments now.
Okay, thank you.
We have one further question from Mr. George Polycladis.
Yeah, okay. I have two questions concerning photoprotection mechanism. The first is why, okay, it seems weird why the plant lets all this energy of the sun go to waste when we have peak sunlight. So the photoprotection does that, okay, restricts, from what I understand, restricts the production of the photosynthesis when we have peak sunlight. It seems like it is calibrated, the energy production of the plant during an average, probably, sunlight during the day, right? Or something
exactly
like that. And the other, is this the reason why the leaves appear to be green? Because
appear what? Sorry?
The color of the leaves appear to be green. I mean, the peak of the sunlight has a peak around 500 nanometers, right? It's green.
Yeah. That's a chlorophyll. The color is due to chlorophyll. Yeah. The pigments that absorb, because the sun energy is absorbed by pigments called chlorophylls, which are in a certain place within these light harvesting complexes. So these are the pigments that absorb the energy and they direct it to the reaction centers. Yeah, but as for the,
I mean, yeah, the green color is supposedly to have the peak, I mean, the highest energy in the spectrum, because in the
blue-red, not the highest.
No, I mean not energy, power. I mean it's the intensity is higher. Because the sunlight has peaked there. It's not the energy, it's the power, right? The intensity.
Yeah. The chlorophyll absorbs this light, and it actually absorbs not the green light, because it emits the green light.
Yeah, why? Why though? I mean, it should be the opposite, right? It should absorb the bandwidth where the intensity is higher, right?
It depends how the photosystem. Okay, the plant is just a short story. So these pigments called chlorophylls or carotenoids are placed in special, let's say, places within the light harvesting complex, so to take advantage the full spectrum of the emitted light. This means that they absorb the energy. We have different kinds of chlorophylls, like chlorophyll B, chlorophyll A. We have different kinds of carotenoids that absorb at different wavelengths. So This means that with different, let's say, pigments, we have an absorption over the whole light spectrum and takes advantage of this. But the plants need to survive under also shady circumstances. Like imagine a plant living only on the full sunlight, and when a cloud passes by, or let's say we have a shade, it dies. So the photosynthetic mechanism has evolved to survive even at low light. This comes at a cost, because if it needs to survive at low light, then it means that the photosynthetic machinery should work also under low light. So it's everything coordinated in order for the excited energy to reach the reaction centers, and then from there to, let's say, to other pathways and synthesize ATP, which is the energy currency of living organisms. But when there is an exposure to high light, then there is an overload of energy reaching the reaction centers. This means that you can produce reactive oxygen species, which is very deleterious for the photosynthetic mechanism. They start to disintegrate the proteins of the photosynthetic machinery. So you need this photoprotective mechanism to, let's say, control the amount of energy reaching the reaction centers. So there is a fine tuning of the amount of energy that should be absorbed at what wavelength should be absorbed and at what intensity and reach the reaction centers. So there is a balance between this, and the excess amount of energy that should be dissipated to protect the photosynthetic machinery.
Right. So it's calibrated for somehow steady power input.
Steady and low light.
Right. Okay. Okay. Then.
Because there is a problem of overexciting the photosystem.
Right. Right. And another question. I mean, for the LHC2, the light harvesting structure, the second one, when you said there is there isn't this mechanism of photoprotection, right? Is this what I understood? Oh, no, I mean,
there is no crystal structure associated with the light harvesting state. There are two conformations of, it's the same protein. It's the same thing here, the same protein, the same antenna, but it has two conformations, like moving atoms in different energy surface, in different minima of the energy surface. So there are the same protein, but let's say some structural changes between the two. And actually, we have no crystal structure yet for this state here.
But they do the same thing, no? Or
yes, okay. Yes.
Okay. Okay, thanks. Beautiful, thanks.
Okay, thanks very much. There's no more questions at the moment.